first. I'm a medical doctor. I'm talking to a lot of medical people here, so I always have to start with a few of my conflicts of interest. Just a few. Suffice it to say, at the top left, I've started a bunch of companies. I consult for a bunch of companies. You probably don't want to believe another word I say over the next 40 minutes. Wouldn't blame you. But I'm most proud of the bottom right of this slide. Those are all the companies started by my students. More than half my graduate students now start companies. And they do this even if they go into academia. And they do this with the most amazing platform there is. It's data. It's often big data. It's often big open data. I'm going to show you how they do it. I'm going to show you how I do it. Maybe I'll convince you to do it. And maybe I'll convince you this is the most amazing time to be in biomedical research. A lot of people use a slide like this as a slide of shame. For me, this is a slide of significance. I think in academia, we're starting to finally learn that if you want to change the world, you just can't keep writing papers about it. You got to take it outside of the university and take it further. So with that, why don't we begin? If you haven't heard, we're in the middle of this data deluge. My favorite example of this is this guy collecting ones and zeros. The Economist magazine 3 years ago pronounced that the human species now generates 2 zettabytes of data every year. If you forget your metric prefixes, it goes kilo, mega, giga, terabyte, petabyte, exabyte, zettabyte. And of course, the following year, it's 4 zettabytes of data because this is doubling every year or two. And I'll be the first to admit most of these zettabytes of data are these YouTube videos of kittens playing with pianos, right? Entertainment value, perhaps, no scientific value, but there's scientific data in the zettabytes as well. So the Large Hadron Collider generated petabytes of data to help those scientists find new subatomic particles. But my favorite example is NASA. So NASA announced about a year ago that by the end of this decade, they will have so many telescopes pointing to the sky that they will be generating an exabyte of data every day, okay, every day. Now, something interesting about NASA's exabytes, though. They already admit that they generate so many pictures of the sky, there are not enough astronomers on this planet to look at them all, okay? So what do? You get your friends on the internet to help. Even NASA crowdsources to citizen scientists. You can sign up at Galaxy Zoo. You can take a test. Is this a star? Is this a galaxy? You can kind of assign what these things are. And it's a telling example that when you run out of scientists, you need to recruit more into the field. And you know I'm going to say we're already there in life science as well. In fact, this is one of my favorite articles. This came out in Wired magazine. Chris Anderson, who was the editor-in-chief of Wired, he announced in 2007 that we have so much scientific data now that science itself is getting obsolete, okay? Now, how do you say science is getting obsolete, right? Oh, actually, it's a scientific method. Remember in 4th grade, 5th grade, you learned a scientific method. You have a question, or we scientists call it a hypothesis, and then you go make measurements to address that question, right? You go gather data. We're already in the world that we already have the data. The new magic, 99% of the work for me and the people who think like me now is figuring out what's the question you want to ask. What's that killer question that everyone's wanted to know the answer to, and no one realizes we already have the data to ask and answer that question, right? That's the hard part. It's not writing the code, it's not cloud computing. What's the question that everyone needs to know the answer to? That's the hard part. And of course, we're there in life science research. We're there because of these high throughput measurements in molecular biology and biochemistry. You've already heard a couple of these technologies being discussed today. My favorite is the one on the left called an Affymetrix gene chip. I got my start with the first 60 of these chips. And the Affymetrix is in Santa Clara, California, kind of near Intel, so they make them look like computer chips. You know, in fact, they're so cheap now that I can carry one in every one of my suits now, and I'm holding one up here in my hand, okay? This is what these chips really look like. Big data comes from small packages, just like this, right? And it's amazing. These chips, you put a sample of tissue, cancer, normal tissue, healthy samples, and you get a readout of the entire genome. Here you're looking at the recipes, the genes, how much is being cooked up from one or the other. You get a readout of the entire genome, a couple $100. And then when you get tired of measuring them one by one, you measure them in the 96-well plate. That's cheap enough now that I can carry that in my briefcase, and I have one of those in my hand here. And then when you get tired of measuring them 96 at a time, you use the 384-well plate, and I have that in my pocket as well, right? I cannot more clearly illustrate exponential growth in biomedicine except to show you these plates. This is what we do. Why even bother doing one by one anymore? You're measuring them 100, now you're measuring them almost 400. This is exponential growth in biomedical data. And it's not just these chips, it's RNA sequencing, it's DNA sequencing, it's mass spectrometry, it's proteomics, it doesn't matter which one it is. There's so much of this data being measured today. Now, something magical happened with these chips. Everyone started using these chips. I got my start with the first 60, and everyone started using them. The price plummeted. Every major academic medical center has these. And what happened in the early 2000, 2001 is everyone started to write papers with these kinds of chips. Here's a list of genes that changed in that cancer. Here's a list of genes that changed in this disease. And you know what happened? The top-tier journals, the medical journals, they said, hold on a second. We're getting too many of these papers with these chips. No more papers with these chips, okay? Unless you put this data into an international repository so other people can double-check the math. It's kind of important to double-check the math now. We're getting closer to the clinic. And maybe others can use these samples for something else. And fast forward to August of 2012. This article in Nature came out, featured my lab, many others. August of 2012, we hit 1 million of these chips publicly available. Okay, you can see the growth chart from 0 to 1 million. Now, if you look really closely at that curve, kind of looks like it's slowing down. It looks kind of sad, right, on the right there? And then you realize they only counted half that year. This is August of 2012. Still doubling every 2 to 3 years. If you were to go to the databases right now, it's about 1.9 million of these chips publicly available. In this 40 minutes, we're going to add another 100 or 110 samples just by doing nothing, right? Now, what does that mean, right? You can get lost in millions and billions and terabytes and petabytes. What does that actually mean? Any scientist studying any disease can go to this website and get samples that have already been run. Maybe it's a rare disease or common disease. Forget the scientists. Any high school kid that needs to do a science fair project now, she can go to these websites. Let's say she's doing a science fair project on breast cancer. She types in breast cancer and search and finds and downloads, you see the circle on the left, nearly 60,000 samples of breast cancer publicly available. She can download about as easily as she can find a song on iTunes today. What is this, right? Samples of some researcher had to submit their data to the repository, and there they are. Now, what's so magical about 60,000 samples of breast cancer, if you haven't figured it out? That's more samples of breast cancer than any breast cancer researcher will ever have in their lab. Because every one of them has to contribute data to the database, right? So whoever figures out the database in the middle has now more data than any one contributor. Maybe it's not breast cancer, it's prostate cancer, diabetes, whatever disease you want. Some researchers probably use these types of measurements or others to study that condition. And all that data is just sitting there, waiting for you. Because nobody else is there. There's not enough people to actually go download this data and do something else with this data. Now, actually, the number I like even better is right above that 59,000, 2445. 2,400 different research groups have already run samples. They already contributed data into this repository. Let me tell you how ridiculous of a number that is, 2,400 groups. Let's say I'm a brand new researcher in breast cancer. I'm a newbie. I know nothing. And I propose to you, let's say I'm writing an NIH grant, you're my review committee. I propose to you that I will get the 2,400 best research labs in the world studying breast cancer to run their best characterized patients on the most advanced measurement technologies that they have. And I propose to you that they will all give me that data for free, okay? You're already chuckling here. You realize when I explain this in a forward direction, this sounds ridiculous, yet here it is, right? There it is, 2,400 labs are there. So when I think of public big data, the term I like to think of is retroactive crowdsourcing. What does that mean? 2,400 labs are there to help you, and those labs don't even know they're helping you. Actually, 2,400 labs are there to help you, and you don't even know they're helping you. And if for some reason 60,000 samples are still not enough for you to get started, just wait another year or two, it'll be 100,000 samples. Just by sitting here, it just continues to increase. What an amazing time to be in biomedical research today. They give you these samples for free. Now, if you have any doubts a high school kid can do it, here is the high school kid that did it. I've yet to meet her. This is Brittany Wenger. I talk about her in all my talks. She's from Sarasota, Florida. She's now a Duke undergrad. And at 17 years of age, as it says on the left, she won the top Google Science Fair competition, top prize for creating an artificial brain, neural networks, deep learning, to diagnose breast cancer. She took immunohistochemistry data and PCR data off the internet and built a diagnostic from that. I guess she was bored, so the following year she did the whole thing again for leukemia, and she was in the paper for that. And of course, what I mean is if a high school kid can do this, we all can do this, and we all should be doing this. In every one of our organizations has anything to do with biomedicine. This is one way to do science today. Now, that's what high school kids can do. What are we going to do with this data? I'm just going to give you a couple stories. My first story starts with this guy. He's a hero of mine. This is Linnaeus. Linnaeus is the guy who actually came up with the binomial nomenclature. Remember how you classify species? Remember kingdom, phylum, class, order, family, genus, species? Remember you all had a test on this at one point. You all had mnemonics for this. Some were clean, some were dirty. Don't repeat them here. Everyone had that on a test. But Linnaeus is also known or actually forgotten about something else too. He was actually the first guy to put diseases into a taxonomical tree as well. The word for this is nosology. It has nothing to do with your nose. Nosology is a systematic classification of diseases, and he's not remembered for this because he got it completely wrong. He said, put all the fevers over here, and the fevers with skin problems over there, and the mental disorders, I'll just put them all in one category down there. He's not remembered because he got a start to this, but he got it completely wrong. But he launched 100 years of the thing to do if you were learned in philosophy was to classify diseases. And that led to what we now use called the International Classification of Disease, or ICD-9, right? We know ICD-9. We just moved to ICD-10, the 10th edition. Actually, it's the 9th and 10th edition of something that started in the 1860s, okay? Just after the American Civil War, the Royal College of Physicians created that system. Now, we can pick on ICD-9 and ICD-10. Let's just show you what ICD-2 looked like, okay? This is the second edition 100 years ago, and you can learn a lot from looking at these old books. ICD-2, 100 years ago, 1909 is when it actually came out. And it's amazing. If you look at these old books, they're written the same way our current code books are written. Use code 39 for cancer of the cheek, the buccal cavity. Use code 40 for stomach, liver, cancer of the intestines, code 41, cancer of the skin, code 44. And then this book does what we still do in code books, even in ICD-10 today. We're running out of numbers, enough with the cancers. Put all the rest of the cancers in 45, right? Not otherwise specified. Lung cancer and brain cancer 100 years ago were not otherwise specified. It wasn't worth even distinguishing between these two, right? This is from the book, right? Isn't lung cancer the #1 cancer killer today? 100 years ago, not even worth distinguishing from brain cancer, right? You learn a lot about medicine by looking at what we put in this bucket called not otherwise specified. It's all the embarrassing stuff we don't know what to do with in medicine, right? You learn a lot in that. And then this code book literally has to say that if you see a patient that died because of visitation from God, be sure to use code 189, right? It's literally in the book, right? Now, same way you're laughing at this. They're all going to be laughing at us like even 20 years from now. Forget about another 100 years. Those silly fools didn't know this bowel disease, this joint disease, same disease, right? Think about how backwards medicine is. We're entering this era of precision medicine. This is imprecise medicine, right? And we actually even make fun of this. We make fun of our transitions to something better, like ICD-10, right? You've seen all the memes out there. This is one I really had to laugh at, We have codes for everything in ICD-10, right? This one, I don't know where this one came from, but this one showed up in my Twitter feed, right? We have a code for getting sucked into a jet engine, okay? Which I guess actually happens on aircraft carriers. Someone actually told me this, right? Right? But why are we making fun of this, right? This is precision, right? What we should be doing is making fun of what we had, ICD-9. This is ICD-9, and let's laugh at that one the same way. Here's ICD-9, the code for lung cancer again, 162.9. Do we have a code for adenocarcinoma, squamous cell carcinoma, small cell cancer, bronchoalveolar carcinoma? No. You know how we distinguish lung cancer in ICD-9? Left lung or right lung, okay? You gotta get that right, but that's imprecise medicine. It's not even like ICD-10 fixes all of this yet, right? Let's not make fun of moving to ICD-10. Let's go beyond ICD-10, right? That's what we got to do to communicate to people what we think this patient actually has. We shouldn't be picking on this. We've got to go beyond this. So how do we go beyond this? And one way to go beyond this is to think about diseases not based on what kind of doctor sees those patients, but based on the molecular profile. So why do we want to do that? Because of this problem with the drug companies. And what do I mean by the drug companies? If I ask most of you, let's say I can actually ask most lay Americans, how much does it cost to develop a new drug? Everyone knows that number, right? It's $1 billion. It takes 10 years is what they say. And actually $1 billion is an underestimate, right? If you actually want to know how much it costs to develop a drug today, it's kind of simple. This article in Forbes comes out every year. It's simple math. Pharma R&D divide by the number of drugs, right? Simple math. How much did you spend? Divide by the number of drugs. And then you realize it's between $4 billion and $12 billion per drug. Kind of not sustainable, right? Now, you may love the pharma industry. You may hate the pharma industry. I'm not here to pass judgment on them. But I'm kind of worried. Where are the new drugs that we need going to come from if it's going in this wrong direction? And then you realize it's going to have to come down to all of us to develop these drugs. We cannot count on the big companies figuring this out. And it's going to be a revolution in the smaller guy and the smaller gal figuring out how to get to these kinds of drugs. And how do we do that? We're going to use public big data to do it. On the left here is just an example. I'm not going to show you genes and double helices. I'm just showing you colors based on genes. And just think about it. My first big grant from NIH is go get every human disease studied by these chips. We get disease samples and healthy, disease and healthy on the left. And while we're collecting those, people start to study drugs. And they put drug data on the internet. Here's some cells with the drug, without a drug, with the drug, without a drug. So on the left, we got with the disease, without the disease. And on the right, we got with the drug, without a drug. The reporters on this called this Match.com for drugs, okay? Got to put these two together. And what's the famous saying? Opposites attract. And let me just show you all the genome methods with my arms. If I got a disease where this gene went up and this gene went down, and I can find a drug that can make this one go down and this one go up, maybe there's a match there, right? That's with 2 genes. Imagine 20,000 genes. I don't need to show you formulas. You get the idea. I'm just looking for drugs that naively reverse what we see in those diseases. And you know what? Let's just turn the crank. And we got lots of ideas. Here's a drug. There's a drug. There's a drug. And where we got a lot of interesting ideas isn't the new drug, but the new use for the old drug. Drug repositioning. Now, we didn't invent this concept of drug repositioning. There are some classic examples. One classic example is a cardiac drug that had an interesting side effect of hair growth. That's minoxidil today, we know that. But of course, everyone knows the other cardiac drug that had its own interesting side effect. Of course, that's Viagra, right? Patients don't want to return that one after the trial. Very few people realize Viagra has done a double switch. There's another branded name called Revatio, same molecule again, but that's specifically indicated for babies with pulmonary hypertension, so the blood vessel from the heart to the lung needs to get expanded as well. So people were cracking jokes about babies on Viagra, right? But it helps with that. But you know what? Instead of finding these by accident, how about we find them on purpose using public data? And that's what we do. We turn our crank. We got lots of ideas. Lots of ideas. Now, how are we going to test these? Well, we got to put them into patients. And all my clinical trials friends run away from me because we got too many of these to try. Well, before you put them in patients, you got to test them in animal models. You might not like animal testing, but we got to do it before you actually try this stuff in humans, right? And all my animal friends, they start to scatter. Well, we have good work with a few of them, but how are we going to get the animal testing actually done? And now when I really, really need a drug tested in an animal model, I go to assaydepot.com. A few of you chuckling, you realize this sounds like Home Depot, and you're right, okay? You just heard from Raymond, right? You get this concept here. Well, these are my aisles: biology, chemistry. DMPK stands for drug metabolism pharmacokinetics. If you don't know what that stands for, don't click there. Pharmacology and toxicology. Well, let's say I need to run a drug study. Let's click on pharmacology. What do you want to run today? What kind of mouse? Bone, cardiovascular, dermatology, diabetes, genitourinary, infectious disease, inflammation, neurology, cancer, eye, ear, pain, respiratory. What kind of mouse do you want to buy? Well, I'm a diabetes guy. Here's an OB diabetes model, 16 mice. Maybe it's 2 groups of 8, 4 groups of 4, your choice. What is this mouse? If you read carefully, it says this mouse has been eating a lot and getting diabetes since 1951. This is a standard mouse model for diabetes, the American way. We eat a lot, we get diabetes, right? And in 28 days, they'll test the fasting blood sugar, the insulin tolerance test, you test your drug, and they give you all those answers back. What am I covering up here? The price, $9,000 for this service, 9-week turnaround time, and literally, you see the button there, add to shopping cart. You can buy an entire mouse model off the internet with a credit card today, right? What an amazing time this is, right? Now, some of you are deeply concerned at this moment, right? All this computer stuff, makes some sense, but this seems like the most important part, right? Because if this works, it's going to humans next, right? How do you trust all this? And some of you with really sharp eyes see this one's run in China. Some of you may have a problem with that, others not. I don't. But actually, when you hit add to shopping cart, you get the 133 companies that offer this service, actually, not just one of them, including Maryland and Wisconsin. So, outsourcing doesn't always mean offshoring. You can pick and choose wherever you want. In fact, there's certifications on the right. Maybe I need them USDA approved. Click on that. I see 4 of them, ISO 9001, 7 of them. You click and choose to get the certifications you want. Now, some of you are still very deeply concerned. How do you trust the results from these companies? And I'll simply say, when I really, really need to know if this drug is going to work in this animal model before going to humans, I just order 2 of them from 2 companies. Hell, at $9,000 each, I can order 10 of them from 10 companies, right? Why am I saying it like that, right? How many biologists, when they get an experiment to work, do they go across the hall to get a completely independent team to repeat the experiment? You know nobody does that, right? It's probably a problem right now. But I can just hire as many independent teams as I want if I have enough money to repeat this experiment. So what I've subtly done there is I've made the quality problem a function of dollars. You want more quality, more dollars. And here's the ridiculous part of the whole story. I don't trust any one of these experiments, but I trust what they show me in common, right? Wisdom of the crowd. I see what's in common. I don't trust any one of these companies, but I trust what they get in common, right? And because of that, now my trust factor is even that much higher. The quality problem goes away because I just have to spend more money and go more independent teams to chase this down. What an amazing time this is to do this. So we typically get one from the West Coast and one from the East Coast. I have nothing against the flyover states, but it's just easier, And we turn our crank, and we came up with our first example. It got us into Wall Street Journal, the front page of Wall Street Journal. We had this idea of the seizure drug. The data told us the seizure drug called topiramate works on Crohn's disease, inflammatory bowel disease. And, we found one company out of Berkeley because they were having a sale literally. We could get a lot of measurements for, same price as the others. And we outsourced one to a company out in Cambridge, Massachusetts. Frickin' expensive, but the only ones in the world who knew how to do a rat colonoscopy. Okay? Not everyone is chuckling, so let me illustrate the geometry involved. This is a colonoscope. This is a rat. Why would you want to know how to do this experiment, right? Find a company to do this one, right? So these are my famous rat colonoscopy videos. On the left, I have the normal rat colon. It's actually still alive when you do this. It's anesthetized. In the middle, it's a rat with the inflammatory bowel disease. You see the redness. Rat all better on the right with the topiramate, the seizure drug, right? 40 megs of video with this. And just to make it clear now, nobody on my campus knew how to do a rat colonoscopy. So I'm not just outsourcing. Yeah, why would you want to learn? I'm not just outsourcing to save money. I'm outsourcing to get the best damn data in the world. Because there's a company in the world that actually knows how to do this, right? It's actually a company that specializes. So we've done a dozen of these. I don't have time to show you all of them, but this is my favorite one. This is a psychiatric drug called imipramine. It's an antidepressant we showed works on small cell lung cancer. Now here, you knock out those 3 genes listed there, and this mouse gets its own lung cancer. You see the nodules on the top left. Humans smoke to get lung cancer. You knock out these 3 genes, it gets lung cancer. This is work done by Joel Dudley, Nadine Chanchan, Julian Sargent, Ana Sri Cordero. Helped us with this. And the mouse on the right is all better with imipramine. Now, imipramine is an antidepressant. It's still in the pharmacy. We don't really use it because we have these serotonin reuptake inhibitors like Prozac. Why don't we use imipramine? Because it's got some side effects. In some people, it makes you really sleepy. And others, it widens your QT intervals and might set you up for an arrhythmia. Actually, neither of those two side effects sound as bad as having lung cancer, 5% survival rate in lung cancer. And cancer is melted away. It's gone in this mouse, right? So why am I showing you this out of the dozens we have? This is my favorite example. Here we start with public data, which people think is valueless, right? Like kittens on videos. We get this computer prediction. We test cell lines in a petri dish. I'm not even going to show you that. We test this mouse. I'm showing you this cool picture. 15 months after the data, after the computation, 15 months, we get ethics, IRB approval. We launch a clinical trial on this one. Humans are dosed on this now. Total cost of this trial, $50,000. Not $1 million, not even $100,000. Now, I don't know if the drug's going to work or not. It's actually, out of those two side effects, believe it or not, the sleepiness is the worst one. Because if your patient told you only have 5 months to live, you don't want to spend them sleeping. I hear that, right? It's actually true. So we might have to search around that molecule for other drugs. So it may work, it may fail, but we got to do more of these. Because we cannot wait for pharmaceutical companies and biotechs to target all these cancers. They can't do it, right? We have to do it ourselves. But the other reason I do is for emails like this. This is an email I got October 2013. I get an email like this every couple weeks now. It's been a while. Hello, my name is, I'm an Italian man, 48 years old, 3 children, a girl, a boy, and a 17-year-old, all with the same wife. On June 5th, they told me I have small cell lung cancer, neuroendocrine tumor. Today I work 75%. I have different mets, liver, kidney, pancreas, and skeletal. I know I will not live longer than a few months, over a year. My family is desperate. Please, I'd like to try this new cure. This is why we're in this business, okay? It's an email like this every week or two. There are plenty of patients that need us to do this, and we cannot keep saying it's going to cost $1 billion or more for each one of these drugs. It's going to take 10 years to do it. That is not what patients want to hear. 2013, this patient is probably gone already, right? That's why we're in this business. That's why all of you have to be in this business, to reach that number of people. The data is there to help you, and no one else is there to do it. That's the time we're in. It's an amazing time, but we've got to help more of these folks. Now, what do you do in Silicon Valley? You got to start a company around this. So this is the first of the 3 companies I'll talk about. This one, you start with the unmet need. You got the public data. We got NIH funding. We spun out a company, one of the incubators outside of Stanford called StartX, and $3.5 million. Now, let me be really clear. $3.5 million Series A, I'm not mentioning $3.5 million to brag about it. $3.5 million to me is the new way science is going to be done outside of academia. You get the experiment to work, you got your papers published, get it out of the university and get it to patients somehow. The science continues in these companies. I don't know if this approach is going to work for real, but now it's funded on private dollars because there's a private interest here, right? I do not believe it's taboo to talk about companies in academia anymore. It better not be. Because this is how we have to get these discoveries to patients. So we have no other choice. So you got to start a company. There's nothing wrong with that in academia. Now the next story is really about diagnostics. If we talked about drugs, let's talk about diagnostics. You've heard about diagnostics, blood tests, right? How do we come up with better blood tests? We start to realize that, you know what, we have all those samples of healthy and control. Can we use those RNA, those gene measurements to help us tell which protein to go chase after? And we turn our crank, we got lots of ideas. This would be a drug for that, and a diagnostic, and this would be a blood test for this cancer. And then we got the idea of this marker might make a great blood test for leukemia, acute myeloid leukemia, not a good leukemia to have. So we go to our cancer center, and we say, do we have a biorepository? Yeah, we got those samples in the freezer. Do we have any serum or plasma on these patients? We can test our computer prediction. And so actually we saved all the cancer cells. We didn't save any of the liquid around the cells. That's called the serum or the plasma. It takes more freezers to do that. I understand. They said, well, we're an academic medical center. Go get the ethics approval. Go put up the posters. And in about a year, you'll have enough samples to test your computer prediction. Now, what does the computer guy do? We just Google for it. Now, find a store like this, Conversant Bio. I see pictures of blood tubes. I'm in the right store. Browse by disease. What do you want to buy today? Bladder cancer, brain cancer, breast cancer. Oh, there's leukemia. Click on that. What exactly do you want to buy? Viable plated cells? Peripheral blood? No. Special processing leukemia blood? No. Oh, there's plasma. Let's click on that. $55 each. How many samples do you want per patient? Add to shopping cart. What is this? These are major hospitals in the United States and all around the world. When that sample gets done and the doctors aren't ordering anything more, it gets on its way to the trash. And it's completely legal to take the stickers off. We got to all agree not to identify these patients, but you can use that for research use. And companies come in and organize all this, like Conversant Bio and others. And the idea here is, we had to play fair. I'm not going to try to figure out who these people are, but this shows up in 48 hours on dry ice in your lab. This is not the hard part either anymore. The samples are just sitting there waiting for you. So we turn our crank. We got lots of ideas. And the one I really chased down is this disease called preeclampsia. Show of hands, how many of you heard of preeclampsia? Okay, it's about half the audience. This is the half the audience usually that watches Downton Abbey because one of the characters dies of this disease. Spoiler alert if you haven't seen it. Sorry about that. That's the only reason Americans even know about eclampsia. Maybe you know people who've had it. I'm just kidding. But preeclampsia is high blood pressure during pregnancy. A lot of people don't know about it. It's still how husbands lose wives around the world. And this was a very annoying disease to me. It falls between the cracks. The obstetricians, I love them, but they say the delivery is secure. Just take the baby out. The mother gets better within 48 hours. But then we pediatricians got to keep this baby alive for months, right? And it falls right between the cracks, so you can see the cost, $20 billion. So we said, let's come up with a diagnostic for this. Linda Liu, Matt Cooper, and Bruce Ling. So we turn our crank, and we have lots of experiments to start with, 266 data sets sitting there waiting for reuse. Whether it's preterm, preeclampsia, hundreds of samples waiting there. And what's the method involved? Just think of a big Venn diagram, right? I don't trust any one of these experiments, but I want to see what's in common across all of them. That's the whole point here. Including, we end up with a marker like this, hemopexin. You can see the pink are the preeclampsia, the green are the healthy pregnant women, and you can see this is one of 15 markers we found that's up in preeclampsia. And you can see that we published a paper, you can find it there, funded by the March of Dimes. And what do? Well, you start a company on this, right? Spin it out, $2 million seed financing. You start with the unmet need, public data available. March of Dimes funded this to get started. We got one of these translational science grants, Spark grant for $50,000. Spin it out, $2 million. Again, I'm not showing you $2 million to brag about it. The $2 million keeps the science going in the company stage. Now, this one is even more amazing to me because we already sold this company 3 months ago. We've already exited, right? Progenity bought this company. We went from public data to acquisition in 24 months, right? Can we do more of these? You bet we can. Inventor happy, investor happy, right? And I'm giving you my secrets because there's so much unmet need out there, right? We cannot do them all myself, right? We cannot. The data's sitting there just waiting for you to just take it apart and come up with diagnostics. We can do more of these, but we need more help to do all of them. And the final big data, you're going to hear a lot about this during this whole week, so I'm not going to spend a lot of time on DNA sequencing. Suffice it to say, we know the Genome Project took $3 billion, took 13 years in factories like that on the left, the Whitehead Institute, and we're heading to USB stick sequencers. We're heading there, okay? First data off of that thing came out just about 2 weeks ago. It's publicly available. Anyone can go start to chase down the data. It's a no-brainer. This is going to impact medicine. Forget about it in my lifetime. Forget about it in this decade, right? It's going to happen in the next couple of years. In fact, Cliff Reed, who used to run Complete Genomics, he was so annoyed a couple of years ago that people kept saying, oh, it's going to be $10,000 by the end of this decade. It's already cheaper than that. He calculated out that by the end of this decade, it'll be $33 to get your genome sequenced. It costs more than that to park here at this hotel. I can tell you for sure, okay? It costs more than, we're at parking lot level genomes now, right? It's a no-brainer. This is going to be in medicine, right? What else do we have to say? So we were lucky we got involved with the first of these kind of genome patients showing up with their genomes. There are a lot of these kinds of stories out there. The one I'm going to talk about in just 1 second is Nicholas Volker. If you're interested in this story, Pulitzer Prize-winning story about the first kid saved by genome sequencing. Go chase this down. It's a great story. This kid was going to die after spending the first 5 years of his life in the hospital. He was just killing off parts of his colon, his intestines. They finally did a Hail Mary pass, sequenced the kid, sequenced the parents, found a mutation. That led to a stem cell transplant. The kid is actually better. He's not out of the woods yet, but he's on a regular diet. He goes to school. He's out of the hospital. This is the poster child for genome sequencing. This is who Francis Collins shows off in Congress. This is why we're actually doing this. And now kids, N of 1 kids, you don't need 10 or 100 kids with the syndrome. You might even be able to figure out with just 1 kid nowadays. It's an amazing story. Of course, cancer and neurological diseases are becoming routine. This is just going to be part and parcel of how we practice medicine, is by getting these kinds of tests performed for cancer and for neurology, especially if we don't understand what's going on neurologically in a child or in a young adult. This is going to be soon to be routine. And what do? You start a company in Silicon Valley. And this is the third of the 3 companies here. And this one's just been on fire. You start with the unmet need. We curated these databases. We get it launched. $74 million in from investors right now, and this is still going strong here because this is just the time to do this. This is the time to get launched, whether it's academically or industrial, really both here. Now let's end out of the molecular role here, right? You're going to hear a lot about wearables. I know you already heard a lot about these types of devices. Show of hands, how many people have a wearable that they're willing to admit? I have one in my pocket. So it's about 10% audience, which is the usual kind of story, it's 10%. I used to laugh at these people, quantified selfers. They measure everything that comes in and out of their body. Measuring things that go into your body kind of sounds rational. Measuring things that come out of your body sounds gross to me. So I used to laugh at these people until December 2012. I come back from a trip to Hawaii. I step on a scale. I'm 247 pounds. And I realized when I hit 250, they can measure me in tons. That's 1/4 of a metric ton. What the hell happened to me? Silicon Valley is too easy. I'm driving everywhere, right? Bought everything Fitbit makes, the scale, the wearable, the steps. I count the steps. The scale actually has done a lot. Lost 50 pounds in the last 2 years, 2.5 years now. If you don't believe me, go look up my TED Talks. You can see what I used to look like, right? And it's called the Hawthorne effect. You just measure it, it gets better, the Hawthorne effect. And it's Weight Watchers rule, so you bite it, you write it. So I write down everything I've eaten for 2 years. It's amazing. All my friends on Facebook know I've lost weight because they've got the social component. You can figure that out. Now here's the interesting part. I go to my doctor, okay? I'm ashamed to say it's not a UCSF doctor, it's one of these others because I still live in Menlo Park. I go to their doctor, it's been written, it's been documented, they spent $1 billion on their electronic health record system. And my doctor's the last person to know I lost all this weight, the most important health intervention ever, right? Because their $1 billion system doesn't talk to my $40 gadget. Can you smell the opportunities here, guys and gals, right? Can you just see what's going to happen? And especially for those of us in medicine, if we don't get with this program, patients are going to cut us out of the loop, okay? The doctor shouldn't be the last one to know about this, right? It's ridiculous today, right? And we're already there. We're already in this problem. It's got to change. And now my data on my patients is going to live in all these repositories, right? All scattered to the winds. We've got to integrate all of that. Another source of amazing data is the hospital charge and cost data. This is one, nobody wants to be on the extremes, and you want to be in the middle on this one. I was literally giving grand rounds at the University of Miami when this came on the front page of the Washington Post, that they charge $166,000 for the same procedures. Jackson Memorial across the street charges $89,000. Right? So I kind of teased them about this on stage. It had just come out that morning. And they said, that's okay. Our quality is better, they say, right, than Jackson Memorial. You can't say that when the quality data is public too. No, you cannot, right? We, in our medical centers, better get our act together on this data before the reporters do. And why is it they have more data analysts than we do, right? Well, how is the public going to respond with all this data coming out there? We got to learn and see, but we got to act on this data. And what's the next big open data? What half of my research group is invested in? Clinical trials data. What's a clinical trial? The most expensive experiment in the world. And half of them fail. And we have this weird culture where that when they fail, we don't even write a paper about it. It's got to change. The EMEA, the European FDA, is making them share their data. We're going to probably get to our FDA. And we're going to do meta-analyses and patient subsetting and precision medicine. We've got a lot to do with clinical trials data. So I'm skating to where the hockey puck is going. I see this one happening. We give out more than 100 trials at this website. Just go sign up for an account and play with raw clinical trials data. It's all open for you. And so much more data just sitting out there. Just so much. Find a logo that you like or actually hate, because some of those are there too. And all this data is just sitting there. Now, in California, we're already ahead of the curve, as usual. I take some pride in being a Californian here. Governor Brown announced a year before Obama talked about precision medicine. Governor Brown helped us launch this initiative for precision medicine. We're putting state money into science. We're launching 2 precision medicine pilots. I'm lucky enough to run this for the state of California, so I'm really proud of the first 2 that we funded. And we're going to do amazing things. And why did I actually move to UCSF? To launch this Institute for Computational Health Sciences. Yeah, we got to slum it out in that brand new building until they build us our brand new building right next to Illumina and the hospital. You couldn't even buy better locationing than that. But really why I'm there is because we're going to put all 5 University of California hospital record systems together. 14 million patients get their care at the University of California, UCSF, Los Angeles, San Diego, Irvine, and Davis. And we're going to put all that together and learn from all of these patients and health systems, a learning healthcare system. So I'm really proud to be building out this Institute for Computational Health Sciences. And last few seconds, I have to admit, coming from Silicon Valley, I'm so used to these kids, and they're annoying kids that start high-value companies in their garages, right? You know, like Apple and HP, or in their dorm rooms, like Yahoo, Google, Facebook. But I fundamentally believe the next Amgen, the next Genentech is going to come from your garage. Because when you go home from this meeting, you got more than 1 million of those samples and every mouse model you want available by credit card. What else do you need to launch a biotech company today? I know it's hard after that. And I know your credit limit might not cover this. That's what angel investors are for, I admit. And I don't mean making meth in your garage. No. But I do mean making the next cancer drug, diabetes drug. All the tools are there waiting for you. So what do I mean by big data and biomedicine? Do I mean it's about algorithms, programmers, cloud computing, databases, high-performance computing, mobile? No, it's about those things, but it's really predicting diseases before they strike, explaining rare diseases that defy experts, finding drugs for diseases that lack attention, making sure we do the right, safe, cost-effective thing for patients, and it's an amazing platform for biomedical innovation and entrepreneurship. I sum all that up with one word, hope. I think patients are depending on this, the doctors are depending on this, and all this data is sitting there waiting for you. If you just want to learn how to do this, it's all just sitting there, waiting for you to just take advantage of this. Can't do any of this work without a long list of collaborators, too numerous to mention. I have to thank UCSF for recruiting me, NIH funding, March of Dimes, JDRF, a lot of different companies and private disease foundations. I always thank my admin and tech staff. I'd never get a career paper out the door without them. I always thank my mentors, Zach Ane, Keith, and Sam for recruiting me to UCSF. And I always thank my wife, Tharangani Deshpande, a molecular biologist, biochemist. She reads every major paper and grant that comes out of my lab. She starts companies with me now. And lets me go all over the world to give talks like this. Thank you very much.
Thanks. I'll just ask you one sort of summary point. What do you think the physicians of today who are training can best integrate this new thinking from quantified self to big data?
Yeah, so the physicians of today are actually learning. A lot of the medical students today do have wearables. You know, we try to put it in their hands, right? Medical students getting genome sequences, getting 23andMe, they're learning sometimes with their own measurements. But the future physician has to know about this. But they're trained in this kind of generation. The real hard part is, what about all the other doctors that are out there? Are they going to learn from CME classes or things, or are they just going to give up? I don't know. I don't have a good answer to that. But they can't just give up, though. We got to do our best, right? That's why you come to these meetings, because when you go back to your own doctors, you got to make sure that they want to learn about this, too. It's important for them to learn. Right? Education's everything here.
That's it. Thanks, Atul. We'll see you at dinner.
Thank you.
Cheers. All right.